Okay, so today's video is going to cover proteins and nucleic acids, the last two of our four macromolecule groups. Okay, so we're going to start with proteins today. So the structure of proteins is pretty complicated, but just to start out, just like the other three, we're going to have carbon and hydrogen, of course, that's what makes it organic. Oxygen as well, and then also nitrogen and sulfur, which makes proteins unique from lipids and carbohydrates. So the monomer unit of a protein is called an amino acid, which I'm sure all of you have heard before. And amino acids are composed of three main parts. Okay, so there's a central carbon, and then it's bonded to an amine group, which is NH2. Okay, that's what we have over here. Carboxyl group, COOH, and then also a side chain, which is just designated as R. It could be one of 20 different configurations. Okay, so like right here, we have CH2SH, and over here, we actually have a carbon ring. Okay, so these amino acids can vary in their side chain from just having perhaps a hydrogen to something as complicated as a ring or two. Okay, so a protein is going to be formed by many amino acids bonded together, and one amino acid bonds to another amino acid through a peptide bond. Okay, so the amine group of one amino acid will bond to the carboxyl group of the next amino acid. Okay, so right here, COOH and then NH2 would form a bond. Okay, so like I said, there are 20 different amino acids. You don't necessarily need to know all these different variations, but it is important for you to know that there are 20 of them. Okay, and they can vary from being nonpolar to being polar, which just means that there are localized charges of positive or negative, just like in water. And there are some that are actually fully charged, which means that, you know, technically they would be, the molecule would be considered an ion, okay, because they either have an extra electron or they're missing an electron. Okay, so like I said, there are 20 different variations, but you do not need to know the names of each of them. And that's pretty much it. Okay, so a protein is going to be made up of a combination of these amino acids. Now, one protein may not necessarily have all 20 of them, but it could have a pretty large variation of amino acids in its structure. Okay, so going further with the structure, a polymer is either called a protein or a polypeptide, which is just a chain of amino acids. Okay, a protein can be several chains of amino acids that have been bonded together. All right, so the amino acids, they're the non-polarity, the polar, as well as the charged ones. They will interact. Okay, they might form, if there's sulfur in one and sulfur in another, they can form a sulfur-sulfur bond. There are many hydrogen bonds that occur because of the polarity between different amino acids. Okay, so a protein is not just the chain of amino acids, but all of the interactions, all of the hydrogen bonding, and all of the covalent bonding that occurs within that structure. Okay, so there are levels of protein structure. The primary structure is just a sequence. Okay, it's like the alphabet, but it's just a sequence of the amino acids in the chain. Secondary shape form, so these are localized interactions, just local shapes that form through hydrogen bonds. Okay, so the important thing here for secondary is that it's hydrogen bonds that are doing the work. They're localized little shapes. So if we look at the entire structure of a polypeptide here, we can see an alpha helix as well as a beta pleated sheet. So these are two examples of those localized shapes that form through hydrogen bonds. Okay, and in a polypeptide, we could have many different alpha helices, we could have many different beta pleated sheets, we could have a combination of the two. Okay, it just depends on the structure. Tertiary is, okay, we were zoomed in here for secondary. Tertiary is we zoom out and we can see the entire structure. So there's additional folding in the polypeptide chain. So it's all the alpha helices, all the beta pleated sheets together, as well as, you know, additional folding on a larger scale. Okay, so more hydrogen bonds are occurring here, in addition to the ones that are already occurring at the secondary structure. But once we get to the tertiary structure, there are covalent bonds that form there as well. And then the quaternary structure is going to involve at least two polypeptide chains that will interact and form the protein. Okay, so functions, energy source, structural material, the delivery of chemical messages, the reception of chemical messages, fighting off bad guys, so fighting pathogens, speeding up chemical reactions, interacting for muscle contraction, oxygen transportation, and many others. Okay, this is just a short list of some of the wide variety of functions that proteins can have. Okay, proteins, they do so many different jobs in the body. Okay, so let's take a closer look. So structural material, so the keratin in your hair and your nails, they provide a more rigid, waterproof structure. Chemical messages, which are hormones that get sent out from your endocrine glands to a receptor cell. That cell will have a receptor, whether that's in the membrane or perhaps inside of the cell. Okay, so those are both going to be proteins. There are hormones that are not proteins, and we'll get to that whenever we talk about hormones in the endocrine chapter. Antibodies help to fight off pathogens. So if your body detects something that should not be there, your immune system will form antibodies that will specifically attack those pathogens, okay, based off of the information that they're giving off in their antigens. Enzymes help to facilitate chemical reactions. They make them go much faster, either building things or breaking things down, okay, but enzymes help, like I said, to facilitate them to make the chemical reactions go much faster than they normally would. Muscle contraction. This is a diagram that we'll look at much more closely whenever we get to the muscular system, but we've got myosin here, we've got actin here, and it's the interaction between these two that will cause your muscles to contract. Okay, there are other proteins like troponin and tropomyosin which help to regulate that process. Oxygen transportation, hemoglobin. So hemoglobin will attach to oxygen in your blood, okay, on your red blood cells, and allow your red blood cells to be able to carry the oxygen to cells that need it. And that brings us to nucleic acids. Okay, so this is our last of the 4 macromolecules. So the structure, again, just like proteins contain carbon, hydrogen, oxygen, and nitrogen, but unlike proteins, contain phosphorus. So the monomer units are called nucleotides. So we see our basic nucleotide structure here, and then a little bit more detailed here with a specific nitrogenous base. Okay, so there are 3 parts to every nucleotide. So the central portion is a sugar, and then off here we have a phosphate group, and over here we have a nitrogenous base. Okay, so the polymers are going to be long chains of these nucleotides, okay, that form being bonded from from the phosphate group to the sugar of the next nucleotide, whether that's RNA or DNA. Okay, so we have DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. Okay, so a nucleotide, like I said, is composed of the sugar. It's either ribose in RNA or deoxyribose in DNA. So the main difference between deoxyribose and ribose is that ribose has all of its oxygens. Deoxyribose has 1 less oxygen. Okay, we have our phosphate group, which is negatively charged, which is actually very important for the structure of DNA, and the nitrogenous base, which can vary from one nucleotide to the next. Okay, so we have 4 different nitrogenous bases, and they're detailed here. So there are what are called pyrimidines, which just means that they have 1 ring, or purines, which means that they have 2 rings. They provide the DNA alphabet. Okay, so we have adenine, thymine, cytosine, and guanine. And a really important feature of these nitrogenous bases is that they are complementary to each other. So thymine will always bond with adenine. Okay, so on one DNA strand, adenine will always be matched with thymine. Okay, same thing with cytosine and guanine. C will always complement G. All right, so our first example of a nucleic acid is DNA, deoxyribonucleic acid. So we have 2 strands in DNA that is different from RNA. So it forms a double helix structure because of the negative charge of these phosphate groups. It forms that spiral. The function of DNA is to hold genetic information. So your DNA is essentially the library for instructions for building proteins in your cells. Okay, so through processes called transcription and translation, this information will be converted into building proteins. Okay, so the complementary characteristics of the nucleotides allow for DNA to be able to replicate itself, but also for transcription to occur, so going from DNA to RNA. So it's really important that A always binds with T and C always binds with G, so that whenever a cell needs to undergo mitosis, through DNA replication, the DNA will essentially unzip, and then nucleotides will come in and match with each of those strands and form 2 brand new, but exactly alike DNA strands. All right, next we have RNA, and RNA is similar to DNA in that it is a strand of nucleotides, but it is much shorter. Okay, so RNA is just going to be a copy of a gene, and a gene is just a really small portion of any given strand of DNA. So some of the main differences, of course, are the single strand, but also uracil is a different nitrogenous base, and it will be used in RNA instead of thymine. So the function of RNA, like I said, is to copy a specific gene and then transport that information to the DNA, from the DNA to ribosomes in order for proteins to be made. Okay, so it's essentially like, the DNA is the library. It has these huge books that are, prohibited from leaving that library. Okay, but you can always go in and maybe in your notebook, write down that information and then take that information to, outside the library. Okay, so that's essentially what's happening here. The nucleus is the library, the DNA are the big books, and then the RNA is like the little note that you take that you can leave the library with, okay? All right, so the central dogma of biology is DNA being used to form RNA, which is used to form protein. So all the information that your body needs is in your DNA, and the purpose of the DNA is to be able to build proteins, which are going to do all of the processes within your body. Okay, and then DNA, due to the complementary characteristics of the nitrogenous bases, is able to replicate itself so that during mitosis, an exact copy of the DNA can be made. Okay, so transcription is the process in which an RNA copy is made of a certain gene. The RNA can leave the nucleus and go to the ribosome, where it undergoes translation, which is where every 3 nucleotides will code for a certain amino acid, okay? And that forms that amino acid chain, which will be formed once the whole RNA strand goes through the ribosome, okay? Okay, and then once translation is over, we have this chain, right? Our primary structure, and then proteins will come in and help this polypeptide chain to take its shape, okay? So that it can then function as a protein. Right, and our final nucleic acid is just kind of, it's like it's not exactly the same as RNA or DNA. And really it's just one altered nucleotide, okay? And it's called ATP, adenosine triphosphate, okay? So we have adenine here and we have 3 phosphates. So that is the main difference here between ATP and a normal nucleotide. So these phosphate groups, okay, especially right here, holds a lot of energy. So this is your body's main storage of energy after cellular respiration. So glucose is used for cellular respiration. Once this phosphate, so glucose will be broken down in order to form ATP from ADP, which essentially is taking the energy from the glucose and putting it into the bond here between the 2nd and the 3rd phosphate. There we go. Okay, so these ATP then can be used for chemical reactions or other processes within your cells. And that is it for our video for today. So hopefully you are able to take this information in and use it for the macromolecule project that is being assigned today as well. Okay, so complete the Google form and then also look at the instructions for the macromolecule project.